Thank you very much. Thanks for the introduction and thanks for having me here. I will talk, as you can see here, something about diversity, biogeography, and the evolution of freshwater gastropods. In the original title, there was also Europe. In the title, there was 200 million years, but I thought a lot of what I'm talking about is very general. Most of the talk will still be about European systems or Europe's development, faunal development, but most of it has a general conclusion, and I thought maybe just expand it a bit. And before we go into the topic itself, just a second. Yeah. It's actually a very good introduction because I just wanted also to say what's important and why I'm talking about all these things. I would like to always have a combined view on things, to do any sort of biodiversity analysis, study extinction crisis, studying diversification dynamics. You need a good taxonomy. You need to understand the species, otherwise it's just an ID. You do not know what you're dealing with. And to me, it's very important to have this combination of topics, to understand the holistic aspect. In my case, it's freshwater gastropod, but actually it doesn't really matter what it is. And so I would call this whole subject quantitative freshwater paleobiology, and it's exactly what you said. We need to understand the past to understand what's happening today, to see where we are going, perhaps. What will you hear today from my side? I will first of all talk about generally freshwater snails, not so much about their biology, but more like what can we do with them in the fossil record? What are they useful tools for, or maybe not? I will give you a brief and rather coarse overview about the global fossil record, something I've been working on the last year or so in time and in space. I will then give an overview mostly centered about European patterns, about the non-uniformity about species diversity in all its aspects, and then come to the topic of why this is. Why is diversity in biogeography and so on, why is this so non-uniform in time and space? And because of their importance, I will focus on long-lived lakes specifically today, but also in the fossil record. So what do freshwater snails tell us? They are ubiquitous. You will find them also in the fossil record basically everywhere. Of course, there is a preservation bias, and we'll come to that, but today they are basically found on every continent, and the same is true in the fossil record. You will find them back to the Cretaceous on every continent, and even on Antarctica you would find some Pliocene remnants of limnates, for example. They have a hard shell, so they have usually quite good potential for being preserved, which is not the case for most other fossils. So you can trace their remnants and can trace the diversity back in time quite a long while. And because a lot of the families, the groups you would find are very specific for specific environments, you can use them in paleoecological reconstruction. You find a specific set of species or genera, and you will know this is more of a lentic system, this is more of a fast-running system or something like this. Maybe even water chemistry. Also, they can be used in the reconstruction of evolutionary lineages if you want to understand what drives morphological evolution. Gastropods, or generally mollusks, are a very nice tool because they are very interesting evolutionary lineages, especially in these long-lived lakes, as we are about to see. And sometimes, particularly when you look at these lineages, they can be useful for biostratigraphy. So it means you can use these to know when you are in time. Most gastropods have a longer time range, so you might know, okay, this is a middle Miocene, this is an early Miocene species. But in most cases, not so much. But some of these evolutionary midpoints, they can be traced back to a very specific time slice. Also, at least in the fossil record, many of the species are endemic. Might be partly, again, a preservation thing. Today, a lot of these freshwater snails are endemic in springs, which we do not have so much in the fossil record, but in the fossil record, it's especially also these lacustrine snails that are restricted to single systems, which again is quite a good prerequisite if you want to study biogeographic pathways. If you want to understand diversification, you need a certain amount of speciation and extinction events, and for this you need basically endemic species. The first fossil species descriptions in the case of freshwater gastropods date back basically to the 18th century, 1760s, yes, 1760s, and most of them are until the 1850s or something from Europe. There are a few then in the 19th century, or quite a lot in the 19th century, also from North America, for example, but most of it was done by European workers in the beginning, then later also Americans. But for example, the whole of the southern continents wasn't studied before the 20th century, basically, with some exceptions maybe. But so we have a massive bias in the southern continents. There might be also some reality that there are not so many long-lived lakes, for example, but certainly we have research bias. We also have the tropics underestimated massively, partly because of this research bias, partly because of lacking infrastructure to kind of see what is there in the outcrops. We do not know even of some outcrops. So we would expect actually that in the tropics there is a high diversity as it is today, but we don't know in many regions. So this is something we have to keep in mind, and whatever we look at, we need to remind ourselves the fossil record is inherently incomplete, especially in freshwater snails, and it is unevenly sampled across space and time. So if we look at the diversity we have, this is all the data of freshwater gastropods I could get on hands. So basically colored after the diversity in single polygons, and you see Europe kind of here, a beacon, and this is simply a research history. On the one hand, France and Romania do have a quite good fossil record, but I'm pretty sure in other regions we have this too, but it is not sampled. So the lack of fossils there is not that there were no fossils, but that we do not know them. And this is like Triassic to Pliocene, so there might be Pleistocene records which we do not cover here. And up here, for example, we have the erosion through the massive ice sheets, so simply there are no deposits left. So this is something to consider when we talk about fossil record. But what do I mean with, what kind of fossil record do we have? How rich is it really? Altogether, it's about 5,100 somewhat species, more than 40 families in almost 500 genera, which is a pretty decent record and compares quite well to the current diversity of freshwater gastropods. But if we think about that, today is just a time slice. I would actually expect for 200 million years maybe 10 times as many species. But again, there the fossil record and the fossil preservation kicks in. So it starts somewhere probably in the Cretaceous, but our entire information about late Paleozoic freshwater snails is based on, I think, something like 7 species for maybe 50 million years, which is not really satisfying, I would say. And most of them have been attributed to modern families. And in some cases, maybe yes. In other cases, if you have a snail this big, slender, no features, can be anything. So I'm not sure. So I based this talk basically on Triassic to Pliocene because there we have more reliable data, let's say. And you see that diversity across these families is quite different. We have here this big pile of hydrobeads, the biggest family through all time. We have a lot of melanopsids, viviparids, planopids, and limnates. These are like the 5 biggest players through Earth history. They all exist since the Cretaceous, at least some of them even probably since the Triassic, or maybe even longer even. To give you an idea what time ranges we're talking about, this is very coarse, of course, but you may know trilobites, ammonites, dinosaurs, mammals, and so on, and they have existed for a decent amount of time, mammals since the mid-Triassic, for example, but freshwater gastropods outcompete them, basically. In the absolute time ranges, there might be a similar age range as the ammonites in numbers. And if we look at Earth history as if it were a 24-hour day. Like Cambrian explosion would be something like maybe 10 o'clock, 10 something. Freshwater gastropods maybe somewhere here, 11 o'clock, compared to mankind. 2 minutes, maybe? Really unimportant. So, in time, the fossil record is also quite non-uniform. Through most of the early Mesozoic, Triassic, Jurassic, not much happens. We have a couple of faunas, and again, this is basically a preservation thing. But on the other hand, freshwater colonization by gastropods was not a single event. This was probably 15 or so separate events, mostly in the Mesozoic. So we have here the slow beginning of several independent colonization events, and we really do not know when these events happened, because the first occurrence of a fossil in freshwater is very likely much predated by its real descendant from marine origins, and we rarely capture these brackish faunas. And when we look at through time and also through these different groups, you see that basically the picture is repeated. We have some diversification in the Cretaceous and then some peak in the Neogene. And there is some reality to this. This is not all preservation, and I will later show you why. How does it look in space? What is it when we look at continental distribution? Again, rather coarse. I will go through these main periods and continental distributions, just to give you an idea of the major players that when they appeared and what happened maybe in parallel. So, for example, in the Triassic, we have the alleged occurrence of these families. These are all modern families. You would all find them very prominent today. Viviparids, as I said, these forms or some of these other groups also may be already there in the Paleozoic, but who knows really. And most of these assignments are based on let's say, fossils that look a bit like the modern ones. And sometimes they put it even in the same genus, which is really problematic, phylogenetically speaking. And we have here a very limited fossil record. We have some forms in Asia, in China basically, a few in North America, in the United States, and maybe something here in India must be somewhere here. And some of these things are also of doubtful age, so we are not sure how old is it really. As I said, there might be some link to Paleozoic forms, but we are not so really sure. And this would, for example, be one of those hydrobia. Basically, it can be a marine species, it could be a terrestrial species. I don't even dare to, I dare call it a gastropod, but that's it. And in the Jurassic, we pick up the pace a bit. We have definitely more diversity, also in a wider range of families, but still the diversity globally is very low. Couple of species, couple of faunas. We have some of the major players appearing. The limnates are very important here, or physids, and all these other groups also. These are known still today. And we have also a bit of a broader distribution. We have first appearances in Europe and also on the southern continent in Australia, which also show there must be some more data somewhere in between. These are not independent colonization events here. Interestingly, during the Mesozoic, just on a side note, the bivalves were more diverse in freshwater usually. Sometimes you have faunas that are either exclusively or at least dominated by bivalves, such as these pseudocardinoids, which do not exist any longer, and this whole package of rock is basically almost 100% bivalves. Somehow they lost importance through the Mesozoic and were outcompeted by gastropods. Today it's, I think, 1,000 somewhat species of bivalves and 5,000 somewhat of gastropod. And in the Mesozoic, diversity was a bit the opposite. But this is just on a side note. In the Cretaceous, we pick up diversification on basically all the continents. We have many more species. We have more species lineages, more faunas. First groups appeared also, very important groups appeared for the first time here, like ampullarids. Melanopsids, definitely an important group. Theorids, many of those also in Asia today. And they were present on all continents now. We have first appearances in South America and in Africa, but still diversity is definitely in the north. There is not much going on in the south. And we have a big peak in terrestrial snails. They also have been around for a quite long time, much longer than the Cretaceous, but we have in the mid-Cretaceous in Myanmar these amber deposits with at least 30 species, of which most of them are terrestrial, and every year they discover new species. So this is one of those Lagerstätten types where you really find exceptional diversity, some of which look alike the modern ones, and some are very peculiar, like these hairy snails. So basically, more or less contemporaneous rise in diversity, but quite certainly not related for the causes. In the Paleogene, we have also, first of all, we have a recovery of the fifth mass extinction event, which hit gastropods in freshwater, at least in Europe, pretty severely. And we have a diversification in Europe and in Asia. You see here the quite increased species numbers. First occurrences of a number of new groups, but not really a lot, and not very diverse ones. Maybe petinelids are an exception. And we have diversification in certain groups, so especially the melanopsids in these Ceratoidea, they pick up. We have a lot of truncatelloids, like the Hydrobiida, picking up, and limnates getting more and more diverse. And these are still today important players. So there are no big, we have diversity centers, but we have no outstanding hotspots, let's say. But overall, we have a better fossil record, building up to what's coming next. Here would be an example of this one recovery fauna in the early Paleocene, somewhere at the border Slovenia, Croatia, Italy, with these very strange things that nobody really knows where they belong. Might be hemicinida or related to melanopsida, but very, very weird morphologies. In the Neogene, we pick up diversity, and we have the all-time peak, basically. So here we have like the highest number of species ever. A peak in Europe, and just to give you some numbers, for a time interval of 20 million years, we have 42% of all the species ever described from the entire fossil record of the world. This tells you something about preservation and research bias. But we also have a high diversity in these long-lived lakes, because we also have a high number of long-lived lakes in the Neogene of Europe. So a lot of this high numbers is also real to some extent. So we know that species diversity is not uniformly distributed. This is not something new, but which might be interesting patterns to look at. First, we'll have a bit of a look on the variation in species composition, because in freshwater gastropods we have quite a nice biogeographic structuring. We will look at species richness patterns, so which relates to the formation of diversity hotspots, and we will also look at differences in the morphospace occupation, so kind of morphological evolution, morphological diversity. And to do so, we will use examples from the European Neogene, simply because there are the best examples, the best known and best studies examples, and a few of those I've looked on by myself. Just to remind you where we are here. So Neogene is from the early Miocene to the end of the Pliocene, something like 20 million years. So not much, but enough to give some interesting results. Let's start with species composition. We have here quite an interesting case of high biogeographic structuring in the Neogene of Europe. Basically, we use this European species data set that goes back to the Miocene, a bit more than 2,000 species, here also including subspecies, because sometimes we have these geographic subspecies, so this might be relevant here, and more than 2,700 localities, plus some basic multivariate statistics, so that we have some quantitative data to go on and make this reconstruction, what might be reasonable boundaries biogeographically. So in the next few slides, I will guide you through these developments starting with the early Miocene. So in this time, we have a lot of brackish systems, especially here in France, Aquitaine Basin, Loire Basin, brackish water molasse here, Brno would be probably somewhere here, and North Sea Basin, and around at the border of these systems might be some freshwater already, but mostly brackish. On the Balkan Peninsula, we have lake faunas evolving, the end of the Miocene, end of the early Miocene, and we have still a rather low degree of provincialism, but still here a very clear cut in the type of faunas. Especially this one kind of is very important for later. In the middle Miocene, we have a bit of a bigger spread across Europe. We have more faunas in the east. We have a spread also here, Balkan Peninsula, lakes, a bit more lakes, more diverse faunas, also faunas in the Aegean region. And we have an increased provincialism, several biogeographic regions, basically similar to before, but here we have some more faunas, and also with the spread of the Perithetian, with the region we have spread of this diversity. And we have some first interesting cases, biogeographically speaking. So this weird limpet kind of thing is probably related to limnaids, and evolved somewhere here, Illyrian region in the early Miocene, and we find it then in the middle Miocene of southern Austria. And there is absolutely no way it could cross this this region. There was a seaway, there was, even if there were no seaway in times, it would not be possible hydrologically. So it might be one of those first examples where we had transport via birds. This is also happening today quite often. In freshwater gastropods, especially the juveniles or sometimes the eggs are transported on the feet or the feathers of the water birds. The late Miocene was definitely the most splendid era, let's say. We had the highest diversity, also linked to the development of the Lake Pannon here, the big one, and the Dacian Basin, both long-lived systems. And we have, in addition, a diversifying fauna on the Balkan Peninsula, and more so also on the Aegean, in the Aegean region, also in Spain. So we have here a bit of diversifying, biogeographically diversifying, let's say. Also, we have the Lagomare fauna, which is a special case. In the very end of the late Miocene, the Messinian salinity crisis, basically most of the Mediterranean Sea dried up, and we had more or less probably some brackish to freshwater puddles here and there, and we have a single fauna similar from the west to the east, from Malaga here to Cyprus and Crete, all the same species, but just for a very short period. And so all this kind of is reflected in a very intricate pattern. We have here subdivisions here, so more and more the situation is getting a bit more complex. And again, interesting cases. Again, one of those surprising immigrants. This is a form that took a while to figure out what it actually is. It is related to Bulinus, which is today a South European to African species group. And this species lineage formed somewhere here in the Balkans and the Middle Miocene, and suddenly we find it in the Turiec Basin in Slovakia. Interesting case, probably again something linked to bird dispersal, because there is no way they could crawl all that way. And on the other hand, an example of the importance of looking at the species accurately. Previously, it has been hypothesized that there must be some link to the Mediterranean region because there is the same species, but these Sakoya forms, but this turned out to be just bad taxonomy. So if you talk about biogeography, it's quite often very important that you know the species. And a lot of those faunas have not been revised, so there is still a lot of work to do. In the Pliocene, we kind of lose Lake Pannon in the very beginning. We have here, the number 14, follow-up system, Lake Slavonia, with these very fancy viviparate lineages that even Darwin already knew and used as an example. And we have the brackish and sometimes freshwater Dacian Basin. So we move a bit to the east and the southeast. We have still quite endemic and diverse faunas here in the Aegean, Anatolian region, in Italy a few, but in France, and single ones here in the Black Sea region. And this is mirrored in an even more intricate biogeographic scheme down to the province level. So here the situation is very complicated. But what happens afterwards? This is just a very quick and dirty graph we produced some while ago. It's basically unpublished, and we just used the relative abundance of families and made a PCA. Probably I wouldn't do this anymore, but it will show you something very important. So in the Neogene, the Miocene and the Pleistocene, you have most faunas being dominated by these xenogastropod things, these Hydrobiida, Melanopsida, maybe a few other families. And very few faunas that are typical with planobids and limnates. Today it looks a bit different. We have most faunas actually here. Most faunas are these short-lived glacial type of faunas. If you dig a puddle, a pond in your garden, 2 weeks later you'll have these species there, because they are in the waters, they disperse very easily. And these are typical of short-lived lakes, while these ones here, they take a time. It's also something ecological. I will come a bit later. And this is something that is also reflected in, or basically links to the ice ages. Let's come a bit back to that later. By the way, here you see also two lakes here, Lake Ohrid and the Caspian Sea, so two modern systems today. Those are typical ancient type faunas, because there you have these hydrobiid-dominated forms. And these are also much older than all the others, and this kind of fits the picture very nicely. Now, what does species richness tell us? There you need to do some, first of all, some methodological step. If you have densely sampled regions like here, you might run into troubles that you may overestimate the real diversity in a region, so you kind of need to standardize. So we reconstructed species richness in an equal area grid, looks then a bit like this, so that you do not duplicate species counts. And then you have the number of species within a single cell. You can do some heat maps, like with Kriging or other sorts of interpolation algorithms to get some idea of how diversity may have been distributed even in between those unsampled regions. And it would look a bit like this. So in the early Miocene, for example, you still have this rather restricted. If you remember the map before, you will have here this Aquitaine basin, here the brackish water molasse, here the North Sea region. But not so much going really on, not really high diversity. Bit more spread, bit more diversity in the middle Miocene. If you remember, we have here these dinorite, these Balkan lakes evolving, rather diverse, some systems here in Hungary and Romania. And then in the late Miocene, we have this massive Lake Pannon, and just not single kind of local endemics or local rich spots, but basically the entire system. 580 species across its entire lifetime. And here the Dacian basin would be, and here some systems along the Black Sea. And the Pleistocene then, here the follow-up system, the Lake Slavonia, Dacian basin here, the Rioni basin in Georgia here, also important ones. And here you don't see it very well, but also orange-colored coasts and Rhodes basins. We have some fluvial lacustrine faunas there, with also some very nice evolutionary lineages. And those kind of exist also into the Pliocene, but otherwise we lose all those rich faunas, simply because we lose all the systems. Basically here the Dacian basin dries up, the Danube kind of fills up most of it. Same here in the Pannonian basin, we have a couple of these interglacial glacial faunas, some of them decently diverse, let's say, and a few potentially long-lived systems maybe here. But otherwise, we have a lot of changes, and this is certainly also a climatic thing, geodynamics and climatics, but we'll come back to that. And the situation today is very similar. We have here some moderately diverse forms, and we have here Lake Ohrid being this long-lived lake. So if we have a look at the extent of the ice sheets, this, let's say, piling of species where we have a number of diverse systems is probably due to the retreat of the ice sheet after the last glacial maximum, where we kind of have the formation of a lot of interconnected lake systems here around the Baltic Sea, southern Sweden, northern Germany, Poland, Baltic, and so on. But still, the number of species is higher locally here in this single, very small, long-lived lake. How does it look like in evolutionary lineages? So when we talk about morphological evolution. So again, I will give you two examples, in this case both from Lake Pennon, but there are several more examples that illustrate morphological evolution in long-lived lakes. And this would be one of those melanopsid lineages. Basically, they all belong to a single species lineage, start with this elongate, slender one. They start to develop rather bulky forms here with keels, about here, and on the other hand, they go into bulky, small morphologies like here. And this happens only in few, I think 2 million years or something like this. And might be related to environmental changes. At least that's the hypothesis right now. And maybe even more fascinating case is these limpet shapes called Valencianasia, or Valencianios, and they kind of go back to a single typical radix. This limnate forms about this big, a centimeter or so, and over time they develop larger apertures, grow bigger, even larger apertures with ribs, maybe for stabilization, who knows. And in the end, they come up to these very weird things. The biggest one is, I think, this big, 12, 13 centimeters, which for a freshwater snail is decent. And they live in deeper water, probably, at least they have been associated with a bit deeper environments. Might be that they lived on mud grounds, and could be also an avoidance to live in deeper water to avoid competition from other species, because diversity was quite high there. Basically, we have here two lineages, one going into more round forms, one into more elongate forms. So now we've seen that there is a variation in species richness, in the biogeography, in the composition. There is a variation in the morphospace occupation. There is variation, but why? I mean, it's not very surprising that this is, but because we know that this is the case. But this is a fundamental problem, actually, and has puzzled scientists for hundreds of years, at least since the dawn of biogeography in the 19th century. But still the question is, what drives diversification in all its aspects? What drives speciation, extinction? What also contributes to migration, perhaps? Is it mainly abiotic factors like climate? Is it biotic, like competition? Is it extrinsic, again, like climate, for example, something from the outside? Is it also influenced by intrinsic constraints of individual species, like genetic or some sort of any sort of constraints based on the species themselves? And are there any factors that are true for all species, or let's say for the majority? Is there something that is important for all groups, independent of time, systematics, geography, and so on? This topic has been researched for dozens of years in literally thousands of papers. Every week there is a new one. And they come up with very different solutions, with very different results, based on the group they're looking at, on the set of parameters they're using, on the time frame they're investigated, and also on the geographic region. So the question is it really such a big problem? Maybe what can we find out from the perspective of freshwater gastropods? So we had a look at diversification dynamics of freshwater gastropods, basically using here the entire fossil record of Europe over 200 million years. So that now we have a bit more species, a bit more than 3,000 species, 5,500 somewhat localities, 25,000 occurrences. So a rather decent data set to assess this issue of diversification. And you see that there is quite a lot of variation in the rates. Here we see the speciation rate, so the number of events per species per million year. Same for the extinction rate here. I will not go so much into the details of what's happening in the Mesozoic because there is a lot of uncertainty and not so good data. But you see already here that there is a peak in the extinction and followed by a peak in the speciation rate at the fifth mass extinction event. So quite likely here we have the extinction event, here we have the recovery event. And then we have kind of a number of smaller peaks in the Neogene which match very nicely the occurrence of these long-lived lakes in the Neogene. But what drives these changes? So we looked at the last 100 million years in this case, because there we have like the most robust data for environmental parameters, but also we have sufficient number of species records to make this reliable. And there is a methodological issue also that is usually overlooked a bit in the literature. If we have a look at a long time interval like this one, do we really expect that a parameter like temperature affects diversification the same way across 100 million years? I doubt that. So what we did is we tried a new approach, dissected kind of the time interval into these 5 shorter time intervals, 20 million years each, still rather long biologically speaking, but definitely more realistic. And as you can see, the match between the rates reconstructed with parameter sets with the original ones is better than if you just look at the 100 million years as a whole. Because simply, I do not believe that there is a single uniform association through time. This is unrealistic simply. And as you can see, there is quite a fluctuation. If you look at individual windows, here, for example, 0 to 20, we look at Neogene plus Quaternary. It's different factors here. It would be topography that be most important here. In this window, it would be diversity dependence. Here again, a different set. And this would be like the 100 million year window. So again, a completely different set, or more or less a weird combination of these factors. So it really depends. You really need to be very careful about what you're looking at and what you can say. So there are important parameters through most of these windows, but there is no single one. So it really depends what you're looking at. So even in gastropods, it depends, not only if you look across all different groups. Maybe another smaller example from basically the same data set, but we go back to the Cretaceous and just have a look at a single peak here. There was maybe this first, or one of the first diversification events in gastropods, and we wanted to know what is happening exactly and why. And we found that, so we're talking here about late Cretaceous somewhere. Here are some nice maps where you can see how did Europe look like at that time. And we found that the rise of the peak, so this first window, is associated with a decrease in the continental area. And the decrease in the continental area matches a rise in the global sea level. And we believe that the rise in the sea level created vast brackish water systems around Europe, which may have been favorable for the diversification of these non-marine groups, might have been a first peak. And then at the end, diversity dependence kicked in here and constrained this peak, so that diversification slowed down again. At least that would be the textbook explanation. Here are some of these very strange morphologies, like this Pergolifera, again a form that has no real extent representative. And interestingly, this case falls together with the Cretaceous terrestrial revolutions, where angiosperms radiated a lot. And they might also have something to do with this, because angiosperms, either as detritus or as in the forms of diatoms, they are an important food resource for gastropods. So maybe something like a prerequisite for diversification. Then one example from North America. Here we have a much lower diversity, also because we have fewer long-lived lakes, very likely, and diversification patterns and diversity patterns look quite different. So we have, for example, very high diversity here in the Cretaceous and the Paleogene. And we have here very distinct peaks in the Jurassic and lower one here in the Cretaceous, and then again single very short peaks here. So the situation is very different. What are the drivers in those cases? If you look at the Mesozoic peaks, it is very clear that this peak kind of links very nicely to the formation of the Bighorn wetlands. So this is maybe the first big freshwater system in the United States. I think it goes into Canada perhaps. And it kind of developed after the retreat of the marine, this embayment, the Sundance Sea. Here we have actually also a lot of dinosaur finds in this area. This was a wetland, so not one big lake. And it matches very nicely this diversification peak. And something similar happens in the Cretaceous with the formation and so the back and forth of the Western Interior Seaway, partly a very big connection between the Arctic and the Caribbean Sea. And probably again there, the first incision of the seaway caused some extinction, and then the back and forth maybe produced some freshwater environments at its margins that kind of sparked again diversification. At least that's the hypothesis. In the Cenozoic, then the situation is different. We have these single peaks, which interestingly link very well to the Miocene and the Eocene climatic optima. But if we have a look at a statistical solution, what are drivers over the whole time interval? Some of them are not significant, in others a constant model, assuming that there is nothing happening through 200 million years, is better. So in this case, I'm inclined to have a look at qualitative matches and not so much or not only at the whole quantitative solution, because you may overlook single very important links, and these links are real. This is not just coincidence. And it also, we check this with good sampling. So let's come back to this slide. Even in the gastropods, it depends if you go to North America, to Europe in different time periods and so on. So there is no solution. I might add one factor here, and that is chance, because quite often I would expect that it really depends on the very system. It depends on the situation, maybe even a single population, a single specimen being in the right moment at the right time, that something happens or does not happen. And we more often underestimate the role of randomness, of mathematical randomness in this figure. Not everything can be explained by factors. But still, if we look at all these things, at all these parameters, these possibilities, there is something we need for all this, and this is time. We need time for speciation, and in the role of, in the case of freshwater environments, we need a system where this time can unfold, and this is the long-lived lakes in this case. We need a number of stable systems. Today there are not too many. It really depends on whom you ask what a long-lived lake is. Some define it as 100,000 years, some say it's 130 at least, like last interglacial. But today it's maybe 10 systems, maybe a few more if you ask other people. In some case we don't know the exact age of these lakes. But in the fossil record we have a few more. And actually it's not so much the age of the lake or the basin, but the evolutionary age of the fauna, because in some cases, in some of these African lakes or Lake Baikal, the fauna was extirpated at some point in time or almost, and it had to basically restart. And so it's more important for the patterns that we observed, how old is the fauna that we have. And just some clarification, quite often you would read ancient lake, and an ancient lake is basically simply a long-lived lake that still exists, but sometimes there is confusion in the literature, so I will stick to the term long-lived lakes, of which we have many more in the fossil record. So in the whole of the Neogene, we'll probably have twice as much as we have today. And one big example is Lake Pennon. We've seen that one already, or some of these evolutionary lineages, but lesser known would be the French Lake Bresse and the Brasov Lake in Romania. In many cases like these two, we do not know exactly how long they existed, but we know that they were there for a pretty long time, so they were long-lived, but we cannot give you a number and use this in analysis, which sometimes makes things a bit more complicated. But still, a lot of the hotspots that we had in the late Cenozoic are linked to long-lived lakes, or at least long-lived systems. Can also be a brackish embayment, like, where is it? The Dacian Basin. Sometimes in the latest Pliocene, early Pleistocene, it was a lake, but before that it was basically mostly a brackish embayment. And most of these hotspots are associated with these long-lived systems, and these are all tightly linked to Europe's geodynamic development and climate. So you need a stable geological basin that sustains a stable lake for an extended period of time, at least hundreds of thousands, optimally millions of years. And you need the climatic stability, like the balance between evaporation, precipitation, that a system can unfold and be stable, or at least that the fauna can be stable for a serious amount of time. So here you see these hotspots and all these red spots. These are tectonically relevant basins, let's say. And the same is for biogeographic relationships. These long-lived lakes also are basically sink systems. They kind of vaccinate the ones around them, and these periparatetian region. So here, for example, we have a look at Lake Pennon here, and this whole region there, kind of, you still see the traces today. The Caspian fauna is basically a remnant of this Parathetis system. The Caspian Sea is basically the rest of the Parathetis. And the same in some of these Balkan faunas. They trace back way longer than we know. And we've seen already the Lake Pennon fauna. Just again to say, it's like 580 species. They did not coexist in the throughout the entire time interval, but still the lake harbored this high number of species over 7.5 million years, which is amazing for freshwater or brackish system. But we have also other examples like these dinorite lakes, which are quite dear to me because there I did my master's and my PhD, and these faunas are so weird and so wonderful because they're so nicely sculptured, and you would find things like this, which is basically an uncoiled planobid. Usually they're planispiral like this, and suddenly you find something like that. What the hell? Or these highly sculptured Theodoxus, which in rare cases are sculptured at all, and very strange forms. I remember when I looked at these fauna at the desk of my supervisor, and I was really, can I please describe this? This is cool stuff. Because it's so rare, you would not find this anywhere else. And basically, the result, these faunas are the result of geodynamics again. So we have here lakes or basins that are kind of oriented northwest-southeast because the Adriatic plate, kind of pushed by Africa to the north, kind of pressed up the whole dinorites. And then we have basins that are highly isolated. We have lakes in most of these basins and faunas that are very isolated. There is basically hardly any connection, and most of those lakes have 90%, 80% endemism. So this is kind of a wonderful island archipel scenario in freshwater. And then a bit later, still in the Middle Miocene, in Serbia, so DLS would be somewhere up there. In Serbia, we have the Serbian lake system with basically the same type of fauna. None of the species is the same with the DLS, but the genera are partly the same, and the type of composition, the relative abundance of these groups is the same. And this is shortly before the existence of Lake Pennon, and we assume, because also a lot of those species groups occur later in Lake Pennon, that this was kind of a stepping stone from the DLS to the SLS to Lake Pennon. So a lot of the things that are traced back to Lake Pennon as their the start point for evolution of some of the lineages that we still find today, actually, they go actually back to this Balkan forms. And from that on, it's hard to trace them back even further. And there we also have these weird, formerly called Kosovia forms. So this is this Bulinus relative I talked about that we had also in the Turiec Basin in Slovakia, and it started somewhere here in the Middle Miocene. Must have come from somewhere, and we really don't know. But to have also a bit of look outside Europe, we have in the Neogene also this huge pebbles wetland which existed for many million years. There were partly a number of lakes, partly interconnected wetland systems with a very, very strange, in this case it's cochleopids, siphonet cochleopids, which is something very odd. You have freshwater corbelids, usually a group that is known for basically brackish, sometimes even marine settings. And another weird examples would be the Lake Nanning, an Oligocene system that is currently being worked on by some colleagues, and also a long-lived lake quite certainly, but the age is not known. We have at least 40 mollusk species, and most important are these unionid ones, but also these huge viviparids. It's this big, 15 centimeters or something like that. And massive shells, so this is a massive carbonate factory basically, and you need to imagine how much effort it needs to build this shell. You even see that there is a very strange cut in the morphology, and this is not in this specimen, this is in all of them. So there must be something going on very interestingly biologically, perhaps high predation, that they try to stick to the ground and have no big opening, and once they are bigger maybe they are not preyed upon it. But very, very strange. But what is happening now? If we have a look in general, but also with respect to long-lived lakes, we know that we are in a big biodiversity crisis, and some even say we are in an incipient sixth mass extinction event. We have here a couple of different drivers. So we have also climate, of course, but it's basically man-made climate change. We have habitat destruction, we have pollution, invasive species, and all these sorts of things. And when we talk about freshwater gastropods and have a look only at the IUCN conservation statuses, and we just predict what happens based on these statuses, even a third of all species may go extinct if we don't do anything. Which would be an extinction rate that is about 1,000 times higher than when the dinosaur disappeared. That should tell us something. And that's not only freshwater gastropods. 2 years ago there was a paper about different groups of mammals, so about the bats, maybe even reptiles, so vertebrates of some sorts, and in all those groups the rates were skyrocketing and several orders of magnitude higher during any of these other mass extinctions. What is when we look at the long-lived lakes? So basically all of those lakes that we have today, they suffer from direct impact of any sort. So we have surface water warming, we have eutrophication associated with that, we have lake level changes, we have pollution in all of them. And this is a critical issue, and I will also show you a bit why. I mean, of course, generally we would lose a lot of information, a lot of species, but we had here specifically a look at the different species groups and how they respond to extinction risk in case of freshwater. So this would be here the Gastropoda on the one hand. They are dioecious, so they have both sexes, they have slow reproductive rates, long life cycles, and the hygrophila, or formerly called pulmonata, they are the exact opposite. They're very fast, and you would find them quickly everywhere. Those are the ones I mentioned in your pond before. And usually they have, because of that, larger distribution ranges. And the question is the extinction risk affected by this? And also, is this a general thing, or is this something that is affected by what we are doing today? So we had a look at this and found indeed that through most of the geological time, with some exceptions, the Gastropoda, so these dioecious forms, they are more often going extinct on a higher percentage, and they have on average the smaller species, the smaller distribution ranges. And if we pool all this data together, independent of time, we see that pulmonates do much better. So they have a higher survival likelihood than Gastropoda. And this result was robust even if you kick out the modern species, if you differently treat these data deficient ones, because you can put them maybe a pessimistic scenario, an optimistic scenario, and it was always basically the same result. And how does this link now to long-lived lakes? In long-lived lakes, the Gastropoda are much more diverse, relatively speaking. So in these, especially in these old lakes, you would have always like 80, even 90%, here in Caspian Sea, 95% dominated by Gastropod. those are also the ones that you would find more often in evolutionary lineages, Valenciennesine accepted. But most of them, the melanopsids, the hydrobates, they do this morphological fancy stuff, let's say, and have these evolutionary lineages, high diversity, but they need time, probably because of their ecology. And this is the result, basically, that you have more xenogastropods in long-lived lakes. So this means, in the end, that these systems are important archives of evolution, especially the ones, these small-ranged species, the small-ranged gastropods, which are usually endemic to these systems. They are massively threatened by what we are doing to these lakes. And the long-lived lakes have been an important source, an important cradle of diversity, and important shapers of diversity, biogeography, and evolution throughout Earth history. And now all these hotspots were linked to this, basically. And today, all those faunas are severely at risk, and we're not only at the risk of losing individual species or genera, but basically a lot of phylogenetic diversity that traces back millions of years. Most of the things that we have kind of trace back to, or many, to Lake Penoman and these kind of scenarios. So to come back to what is probably my favorite slide here, we can add here the long-lived lakes to this list. And what do we learn now from this complicated picture that it really depends where we look at, and there is no clear scenario? Let's bring in a new term. Ecological opportunity is usually used in the context of adaptive radiation. So if you're in a lake, or basically any setting, you need niche space, you need resource availability so that radiation can happen in the first place. But we can scale up this concept, and we call it scale independent. So if we go on a continental, on a global scale, we need, in case of freshwater systems, freshwater faunas, we need the geological system. We need the geological stability, the geodynamic system, the climate stability, that we have a system that we can have a fauna in the first place. Maybe something to think about. Thank you very much. Oh. I suppose, yeah. Or is it different? The Lake Aral fauna, indeed, we did not include this in our study. There were some remnants, I think, in the north that were not completely hypersaline, where there might be some species left, but basically it's an offshoot of the Caspian fauna, more or less the same species, or let's see, the same type of fauna. But as I know, it is completely lost now. Yes. I don't know, also because the taxonomy of a lot of those species group is not settled. We looked into the hydrobiates of that area, and I think there are more than 100 species described, and I do not believe that. Taxonomy is a bit difficult there, and you have a lot of these Caspio-hydrobia things, I think 30 species names, and maybe they might be all just a single species of Acrobia. So it's really difficult to say what we lose. Also there we need to have first some taxonomy, but it's difficult to find living specimens to do genetics. A lot of these older stuff is in dried up or formalin, and it's very difficult to do even ancient DNA. Mm-hmm. So I was wondering if such similar unusual dispersal works may have caused It is difficult, to be honest. And it is difficult to prove because the fossil record of birds is so poor. You have maybe 1,000 finds through 50 million years as a whole. Sometimes it's the only reasonable explanation that that we could find to explain dispersal across watersheds and across mountain ranges and things like this. I definitely believe that if it is possible to establish a population there and not just have like a random occurrence, which anyway you might probably not find in the fossil record, that this has an effect on the fauna. Whether this species becomes invasive in a modern sense, that's a different question. It may be just a non-native species, but I think it depends on who you ask what is an invasive species. But I think usually invasive it is called if it causes any sort of trouble and affects the other species. And that is really difficult to tell in the fossil record because you need stratified assessment on a basically on a ecologically relevant scale, and you never have that, even in the well-resolved lakes. Oh yes, definitely. So if you're in some nicely calcareous alkaline setting, the preservation potential is rather high if you have high carbonate concentration. But quite often we have also these paleostrine settings. Sometimes they're a bit more acidic, or there is root formation in these shallow water regions that destroys a lot of the fossils. Then you know there were fossils because you find the remnants of the shells, but quite often they're crappily preserved. And in really acidic environments, you will rarely find them at all. Maybe some swamps, or so sometimes you have some species that go there, but you barely would find them fossilized. That's actually also a very tricky thing with the land snails, because quite often the land snails are either in loess deposits or actually preserved in freshwater environments, because if you have a land snail on the ground, you will not find it in the fossil record. And so our knowledge about the land snail record is even worse, unfortunately, than for the freshwater ones. I think today is like 30,000 species of land snails, and we have maybe 2,000 or 3,000 fossil ones. It's really bad. For what processes specifically? What do you mean?
Community shaping. So you were talking about abiotic drivers. This is stochastic process, but there are ways to
Yes.
that. Did you try to do this?
Well, in this case, I mean, basically, the method that we chose is a mathematical model, Bayesian mathematical model, but it's not for single communities, because we kind of chose the entire fauna over a long time interval. So there are it's difficult to estimate the true connections between these faunas. If you would have a well-sampled system, maybe with some time slices that are decently well sampled, then we could try that. But there we always run into the trouble of preservation. This Bayesian model is more like on a larger scale, and tries to incorporate also this preservation issue. Kind of it includes a model for how likely it is, based on the number of records in a specific time interval, that you actually captured the real diversity or not. But this is everything that we could do at this moment, simply because the data is not good enough.
You talk about taxonomy, which is like crucial thing, and I want to ask, because in a modern communities or modern biota, you're facing also problems with taxonomy, but we have a tool, we have a easily easy way how to control it or test it using local methods. And when we did it for, let's say, assumingly well-known group, that we found that like 50% of species are okay, and the other half is just bad. And you have all kinds of biases, overlapping, over splitting, or wrongly defined species. And what was interesting, what we find with Jeff, there was a spatial bias that at local scale, people tend to describe more species. They oversplit because they treat themselves to be experts. So like the single difference is very important, but on the big scales, they're more reluctant and back up, so they were more lumpus. So maybe this is something that could be also tested in the fossil record, because for the fossil record, you know, I'm thinking how to define the boundary that you decide, okay, this is a new species, and then going from the literature in like groups that I'm working with, I saw is especially like the biogeographic bias in terms of researchers' mindset. Like in some countries, people tend to oversplit. There was, you know, more species at a given time than the other countries.
Yeah, this is definitely definitely a big problem. Also, this oversplitting in some regions, or this, let's say, uneven research approach, makes it difficult. Like for the Caspian Sea, it was Russian colleagues in the mostly second half of the 20th century that worked on or established most of the species, Starabogato, for example, and they, I mean, there are catalogs about his species, and I'm not sure if 90% are valid, because there are simply different concepts at the time, and I think you were also paid in the number of species or something like this. So it really depends whom you ask and where you go. And I know that what we show here is not absolute truth, that this is of course depending on taxonomy, and if we would have the time to go to all these faunas and check them, the picture might look a bit different, but I still believe it is not so different because there are so different groups in these regions, and that the endemism is so high that a few changes in the status of a species or as a synonym or not would not change the grand picture. At least that's my take on this. It's a bit like with these. Yes, yeah, oh yeah, yeah.
Open up this topic mainly for the audience, and yeah, another thought in this line related. What's the like the age of the species? The evolution is the continuous process. So, like looking at the species right now, you're mostly dealing with some, you know, evolutionary shaped entities. Of course, there might be evolution running, you know, where hybridization is going on. But in a fossil, it's a continuous process. So one time you have to say, okay, here is the boundary.
Yes.
Probably the fossil record helps you because you have gaps and then you see the jumps.
Exactly, that is sometimes the good thing about the fossils, that you lack the transition, otherwise you never know where to do the cutoff. But I talked with a colleague about this, that it really depends also on the species concept, of course, that you apply, and we came up with a very nice new species concept. It depends. It really depends on the system. In an evolutionary lineage like we had with this Melanopsis and Valenciennaceae, you could probably say this is one species because you never can really do the cutoff if you see the whole transition, which is unrealistic. A phylogenetician would probably say, how can you give them even more than one genus name? Because you definitely know it must come from that genus. Why do you give it a different genus name? But then basically everything, every biota on this planet would be one genus, if you think about it. So it is really difficult where to make the cutoff, and it is more or less a probability thing. If you can separate it on a sufficient number of characters in an evolutionary lineage, for example, if you find a cutoff of some way, then it makes sense to give it a name, even if it might not be biologically realistical. And in other cases, the situation might be that you have two systems, parallel evolution of the same species, but based on the chemical or other properties of the systems, the species look slightly different. And paleontologists would always call them two different species, because how could you know? And we can only try to have a broad mindset of knowing about these things, because we can study this in the modern systems, but at some point that's what we have. And then we can only speak in probabilities, basically.
Going back to this last issue. Well, are there cases where you can construct basically a split of one species into two in the fossil record? Which would be, I mean, which would be the classical thing, bifurcation,
Yes.
like that. You can say, okay, this was one species, now we have two others. That's the theory, at least. That a species split into two. If it behaves decently, according to phylogenetic rules.
I think so.
Prove that in a fossil record.
Proof is a difficult word here, but I know of studies that have attempted this. I remember a very weird one that even tried to show that morphology makes this and then makes that again, which in species terms is a bit difficult. But definitely there are examples where there is good evidence for a splitting, at least a morphological split, but this could also be that there is some adaptation, some local adaptation to some microhabitat, and if you put them back together they would still be able to mate or something like this, but who knows. But the examples are limited, I'm afraid. But for me, one interesting case would be these Gyraulus from the Steinheim Basin, this one of these first examples of morphological evolution, and there's some people who say there's 50 species and some say that's 5, and where to draw the boundary. And then there is an example, like there was one that is corkscrew-shaped Gyraulus, which is very weird, and they recently found in a Tibetan plateau Gyraulus lineage, same genus, a corkscrew morphology within the same species, which is just an aberrant morphology with a gene switched on or off. This can be the case in that lineage, but not necessarily means it works also in the other case. So again, it's more or less a probability thing, and if I have more of those, then I would assume there is a driver of this morphology, and if it's just a single one, I would expect there's some random chance. Yes. I'm not sure if it's less. It depends on where you go. You have, in some systems, you still have a high diversity. There are some long-lived lake faunas in Africa and Asia, for example, with high morphological diversity, spiny shells, and so on. But I would also assume that it depends on the availability of suitable systems, because all those, or at least most of those, huge morphospace explosions, let's call it like this, are associated with this long-lived lakes, that you have a, let's say, more or less stable system. That doesn't mean that nothing changes ecologically, but that the system can and the fauna can exist for a long time. All of these developments are linked to time. And then maybe there are local adaptations to predation, like in some of these Tanganyika, Lavigeria snails. But yeah, I think today we don't have as many systems simply as in the fossil record, I suppose. Or there are other constraints or other groups that fill that space. It really depends on the ecological setting and the pressures. They don't do that for fun, I suppose. Yes.
Some layer here for a couple thousand years or something, there
think we try to see average species longevity ranges, and they were quite variable depending on the group. There are usually this kind of, in the entire fossil record, 10, 20 dustbin genera and species that you throw in everything that looks similar. That I would not consider, but some species may exist for 10 million years and others for just 100,000 years. But I don't know if somebody ever quantified that. For freshwater, I think there is something for gastropods and bivalves in the marine realm. I think Stanley did something there. But they came up also with, I would say, more or less random numbers, because they said maybe 10 million years for the average bivalve. But what does it really tell? Did they just have very coarse bins? Or, because quite often you don't know the stratigraphic horizon well enough to tell the exact age. And then it really depends on the taxonomy. So I don't really dare to say a reasonable number there. As usual. That is a good question, and there are a couple of different methods. Sometimes it is biostratigraphy, that you have other marker species, but this usually works best in marine settings, or sometimes terrestrial settings. Sometimes you have quite good magnetics, paleomagnetics, that you can kind of have a, it's also relative, but still you have maybe a good tie of your section, if it's long enough, to figure out where does it fit best. Optimally paired also with some absolute dating. So if you have some volcanic ashes, for example, or some zircon finds in your strata, then you can do some isotope dating, uranium or calcium, argon, or whatever kind of dating possibilities are there. And then optimally you have a basic basis and a top that you can date, and in between maybe some paleomagnetics that you can kind of link what is happening when and how fast, because sedimentation rate is of course very different sometimes. But quite often, yes, quite often we don't know exactly, and then we only know that this is lower Badenian, like this. And it can be anything between a few 100,000 years or a million-year range. It's even worse if you go to the Mesozoic. Sometimes you only know Lower Jurassic. I think that's 30 million years. Okay. Yeah.
Huge difference. But in the past, we were doing better, but we lost them. So
I don't really know, to be honest. The North American fossil record there is not really good, but I had a look on the bivalves as well. But the biggest diversity was in Asia, China, Russia. They had these weird groups in the Mesozoic, these trigonioidids, that somehow distantly linked to marine groups, and pseudocardinoids, something that is more related maybe to spheroidae on a larger scale. But even in the Cenozoic, I think the centers are in Asia, and there is still a center of diversity there. Mekong River, for example, these regions. But in North America, I actually don't remember having this big diversity. I need to look that up. In North America or altogether? I think even there were a couple of hundred species. Yeah, yeah. Even I think the Chinese described a lot from Yunnan, for example, Sichuan, a couple of hundred species in single provinces even. In the Mesozoic or beginning in the Triassic, I think the this part of Asia was terrestrialized. Everything before that was basically marine deposits, and then we had there huge basins like the Sichuan Basin. There was through the entire Jurassic Cretaceous, and then also a bit later we had long-lived lake systems, which interestingly were more diverse with the Bibles. And also in Yunnan there was big lake systems. Yeah, but yeah. Yes. Yeah. Yes. That's the difference there. Yeah, but the problem is there that they are not well preserved. So I would assume that. Yeah. Yes. Right. Maybe the Bible specialized more on the rivers while the gastropods stick to the lakes. Yeah. Yeah. Yeah, that could have been more beneficial maybe for their ecology.